I'd like to thank you for coming to this panel and the organizers for inviting me. Hesperos is a small biotech that is simulating human bodies using actual human cells and also creating computer programs of those systems to predict clinical trial outcomes. So Hesperos was formed by Mike Schuler and myself. He's a professor at Cornell. I'm a professor at UCF. It was spun out about 4 years ago. We've licensed about 20 patents from UCF and Cornell. We won the Lush Prize for alternatives to animal testing. We've won a number of SBIR grants to automate the systems. We started off in UCF incubator and have graduated last year into a 14,000 square foot facility. We have about 27 people in the company at the moment. So the main thing we're trying to do is to simulate what happens with a human being, but not have cell death. In a clinical trial, you really don't want to have cell death. So we do what is called clinically relevant functional readouts. So when you go into a doctor's office, he doesn't immediately then stick you in the arm and be able to measure biomarkers or things in your blood. He'll say, how are you walking? Are you talking? Do you make sense? He listens to your heart. He whacks your knee. He's actually measuring functional readouts. He's looking at muscle contraction, electrical activity. The neuromuscular junction is looking at the reflexes. And these are all things we try and simulate in our human-based functional models. Allows us to not only look at acute, but also chronic administration of drugs in these systems. It also allows us to eventually look at target identification in our phenotypic models. Those of you who aren't familiar with this technology at all, normally in a clinical trial, if you have a drug, you create a computer program. You put a lot of information into it from your experiments, from previous clinical trials, from the literature, to try to figure out what would be a good concentration for a phase 1 clinical trial that won't kill somebody. Well, Mike Schuler about 25 years ago said I could take all these equations, modify them, and maybe use it to design a body on a chip. We now have taken that into, this is a more advanced version of it right now. We're funded by the HEAL initiative to look at opioid addiction, where we can put in multiple organs on that chip. Another thing that's also very important to what we're doing, we have a pumpless system. So the whole thing just fits in the palm of your hand because we use just rocking and gravity to pump things back and forth. It's a very busy slide, very technical, but I just wanted to go through the whole idea of how do you actually fabricate a functional based system. So what we can actually do, we can make surface chemistry patterns on top of microelectrodes, put cardiac myocytes on top of that. We can then measure rhythm generation, or basically your heartbeat, your conduction velocity, electrical activity in the heart, and then the AP length, or the QT interval, which is the length of an individual beat. We can also measure force. We can actually make a MEMS device where we can have a thin piece of silicon here. We can grow the cardiomyocytes on it. When they contract, they bend it. They then have it deflect a laser beam off the bottom, so I know the amount of the bending I can actually get the force. We've measured over 50 drugs in these systems. I'm just going to look at 3 just to prove a point, show a point, which is norepinephrine will generally speed up your heart. We can actually show it does speed up the heart in the dose response manner. Verapamil is it blocks channels in your heart that is responsible for force. You see verapamil right here, which is decreasing the force. Now, Sodalol causes a QT elongation of a single axis potential. The problem is if this gets too long, it interferes with the next axis potential, and it causes fibrillation, and that's where the term comes from. But it doesn't cause any change in force. So we can actually do mechanistic aspects of it in terms of any pathway inside a cell having to do with force. I know it's not affected by this drug, but it is affecting the electrical activity. I can then have systems, we've actually built systems to be able to measure the force, to measure the electrical activity, put it together so we actually can flow a serum-free medium, like a blood mimic in these systems. Okay, and now actually this is, I want to walk you through the whole idea of how we simulate things and why we do it. We did a partnership with AstraZeneca to be able to look at a drug they couldn't understand where the toxicity was coming from. So we made a very simple heart. So this is the electrical activity of the heart, the force activity of the heart, and then the liver here. And the point is the drug terfenadine, or a validation drug terfenadine, which was pulled off the market years ago because it causes a QT elongation. The cardiac had a very large, the compound had a very large effect on the cardiac, but when I added a liver to convert the terfenadine into its metabolite, hexafenadine, which is not toxic, you saw that shifted to the right. The conduction velocity was not affected if the liver was present, but without the liver, you saw a IC50 of about 32 micromolar. But again, cardiac viability is not affected in the system. We're only looking at the functional readout. It's not causing cell death. But then we can then actually simulate the organ on a chip or the body on a chip using a modeling program that we've created. We can actually then look at the mixing. We can look at the loss of, say, absorbing of some of the compound or some of the conversion of the metabolite. So we can get this area under the curve or the pharmacodynamics of the system and be able to then actually look and see what would be the exposure of that compound, okay, of a particular organ in a particular period of time. We can then build a simulation of the system and be able to look at the loss due to absorption into the system, loss due conversion of the liver, to be able to calculate the pharmacokinetics. And then you can look at that, then see how much that affects the pharmacodynamics on the cardiac in the system. You can actually see here the pharmacodynamics shifted, and actually look at the QT interval in that system after it crosses the cell membrane and be able to predict that. Now, because we can actually create a PKPD model simulation of our chip-based system, we can then use that. We figured out how to extrapolate that to then predict what goes on in vivo. And in collaboration with AstraZeneca and a paper that was published in Nature Scientific Reports, we were able to predict QT elongation for guinea pig, dog, and non-human primates, okay, with a pharmaceutical company. So again, we can take our simulation, our experimental simulation system of the human body, create a computer simulation of it, and be able to use it not only to predict, understand what is actually happening in the body on a chip or the human on a chip, but even extrapolate what's actually happening in a clinical trial outcome. We can actually then combine this together with other organs like muscle and neuronal and be able to model that. We published this in Nature Scientific Reports, Advanced Functional Materials. In Advanced Functional Materials, we took it out for 28 days, which is a very important time for toxicologists because that's normally when they run for experiments in rodents to be able to look at systemic toxicity. We've been able to add an immune component to it. This is very important. We're doing COVID research right now with this because we've been able to reproduce the innate immune system on a chip. This was actually published in Advanced Science this year. And these are actually monocytes flowing back and forth in this multi-organ chip where we can actually reproduce what happens with your body when a pathogen comes in that's unknown to the body. It activates the innate immune system. We were able to reproduce both types of monocytes, the ones that actually are protective and causes an inflammatory response, but also the ones that are sort of restorative, okay, in the system. And then lastly, we're able to reproduce and add to these systems barrier tissues. Skin's a barrier tissue. We worked with L'Oreal for a year to be able to work with that, be able to evaluate cosmetics. Kidney as well. Blood-brain barrier separates your CNS from the rest of your body. And then GI tract to be able to look and see what absorbs across the GI tract into your systems. And then lastly, you say, well, okay, what can I do with these systems? What does the FDA think about this? Well, we anticipated that, and we started running workshops through the NIH. I co-chaired with program managers at NIH with FDA, EMA, and pharmaceutical companies, and also other stakeholders in the system. Well, we came up with this whole idea of being able to have preclinical results steer limit clinical trial construction. Basically have your companion diagnostics. You figure them out even before you start doing animal experiments, even if you have to do animal experiments. Limit the number of people for phase 3 clinical trials. They did Vioxx, it was a 30,000 person clinical trial, and still missed some genetic variations. In vitro clinical trials for rare diseases, there's 7,700 rare diseases. At this point, there's only active research programs in 400 of them because they don't have animal models for them. Where we've actually been told if you can actually take, and we're actually working with rare diseases right now, that we can take a patient's stem cells, differentiate out the organs that are really affected by the rare disease. And if we can actually show a compound will reverse that in terms of the phenotypic model that they actually will accept this data in lieu of an animal model. Better evaluations for children, better evaluations for aged individuals, and also personalized medicine. We had a paper recently published in Science Translational Medicine. We're able to show we could actually take and do therapeutic index for a individual cancer in terms of not only its efficacy when exposed to chemotherapeutics, but also the off-target toxicity. What is the toxin in the other organs? Because there's some debate in the community that after you've gone through your third round of chemotherapy, whether that's the chemotherapy that's actually killing you or the cancer. I'd like to thank you for your time. And to all the folks at Hesperos and also collaborators at UCF, I'd like to thank them and welcome to your questions during the panel discussion.